Where is everybody? Oh, where's Mark Twain Lake?
It's by Hannibal.
Oh, okay. So over east.
Yeah.
Okay. I'm unfamiliar with Mark Twain Lake. Is it a nice lake?
Yeah, it was pretty busy this weekend with it being Labor Day, and they have like a little parade thing down there. A lot of people are fishing. It's a really good fishing lake.
Okay. Yeah. I don't know. It seems like a lot of the lakes around here are very cloudy because of all the sediment and stuff.
Mm-hmm. Yeah.
But I've actually never been to Hannibal, really. I drove through there once coming back from New York, but I didn't really stop except to get gas. But I should go there. The birth of Mark Twain and all that.
Yeah. Yep.
Hello.
How's everybody doing today?
What is it?
Did you guys do anything interesting over Labor Day? Mary's like, no, I just stayed in this room the entire time.
We smoked a bunch of stuff and had a lunch with our family.
Like smoked meat? What kind of meat do you smoke?
We made some pulled pork, some brats, and then my nephew helped me grill some hot dogs.
Okay. So when I moved to Kansas City, I, of course, heard about Kansas City barbecue. And coming from Texas, barbecuing is a big thing, but it varies quite a lot geographically. And being from Texas, barbecuing is all about slow cooking your meat and smoking your meat. And Kansas City barbecue, I think, is more about grilling your meat and then slathering it with barbecue sauce. And so I was not a big fan of Kansas City barbecue when I moved here, and offended a number of people as a result of it. And then my partner, who is from the Kansas City area, never really liked barbecue. And then we had barbecue down in Texas, and she's like, "Oh, this is really good." I'm like, "Yeah, it's just difference in the preparation." So I'm a slow smoking kind of person.
Yeah, the barbecue's kind of a point of pride for people.
Yeah, certainly. So what about the rest of you? Did you have any interesting food over the weekend?
I went down to Springfield, and there's this restaurant called Lambert's. There's only 3, I believe. And
the home of the throat rolls. Yes.
Yes, it was very, very good. The rolls are good. The rolls are very, very, very big, and I didn't know they were that big.
Did they throw them at you from across the room?
Probably about 6 or 7 feet away, not like all the way across the room.
Okay. Yeah, I've been to Lambert's once. Well,
Still
we
a
made, what was that, Marcus?
I would say it's still a great experience. It was fun.
Good, good. We made Asian dumplings and spring rolls over the weekend. This past spring, for some reason, I decided, well, okay, mainly, so have any of you guys been to the Blue Koi down in Kansas City on 39th Street? Excellent Chinese food, probably my favorite restaurant in all of Kansas City. And I like their black bean tofu, which is really good, but they also make dumplings. And then when the COVID crisis came and everybody shut down, I could not get my Blue Koi fix. So I had to learn how to make Asian dumplings myself. And the nice thing about them is you can make about 40 dumplings out of one batch of dough. And you can freeze them before you cook them. And then whenever you want dumplings, you just pull them out of the freezer, thaw them out, and cook them up. And then they're just ready to go for you. So I made a big batch of dumplings and had dumplings the last 2 nights. And spring rolls, which I also like a lot. And that was fun. Mainly labor saving for the future. So how's the food blogging going and the food journaling going? Is everybody squared away on that now? Okay. All right then. Any questions about photosynthesis that we finished up on Friday? So the things that you need to know, just to remind you, are things like being able to distinguish the difference between C3 and C4 photosynthetic pathways. What does that mean for energy efficiency of photosynthesis, and what does that mean for water balance of the plants that do those 2 types of photosynthesis? And then how those 2 different forms of photosynthesis then affect where you might be most successful at growing those plants in terms of the growing climate that those plants are found in. And so if you can do those things, then you should be in pretty good shape. So today we're going to start talking about genetics so that we can start talking about genetic engineering. In the Omnivore's Dilemma, Michael Pollan talks about corn genetics once again here in chapter 1. If you look at the section on the rise of ZMAs, this is section 3 of chapter 1. He begins talking about section 3 and section 4, talking about the genetics of corn, corn reproduction, corn sex. And so the latter half of this chapter is all about things having to do with reproduction of plants and the genetics of plants. And so once again, in order to understand this material here at the end of chapter 1, we have to once again go back and get some basic science. And then we're going to finish up at the end of next week with genetic engineering, which once again, in order to understand that, we need to understand the basics of meiosis and genetics and all of that. So the PowerPoint slides that are on the Moodle site they're going to cover multiple days because this is all kind of a unified unit of information. We're going to split it up into different sections, but once again, it's a multi-day endeavor. So let me pull those up. Where did they go? Here we go. So I'm going to share my screen with you. Well, why is the sharing screen thing always so difficult? All right, let's try this now. Okay. Can you guys see the PowerPoint?
Yes.
Okay, cool. All right, so we're going to begin talking about flower structure, just so you kind of understand what the reproductive parts of plants are. Then we're going to talk about the cell cycle and mitosis and meiosis. Oh, and that's supposed to be central dogma. And then we're going to talk about this thing called the central dogma, which is how you get from genes to the phenotype, what organisms look like and behave like, and things like that. And then we're going to talk about genetics. But today, my goal is just to get us through mitosis and meiosis. So this is a gigantic slideshow. It's got 53 slides in it, but we're only going to be going through a short portion of that today. So kind of learning goals for today are, you should be able to look at a photo or a drawing of a flower and know the parts of the flower that are involved in reproduction. Be able to identify the different phases of the cell cycle. That probably doesn't mean anything to you right now, but it will here in a little while. It's just the phases that a cell goes through as it grows and then ultimately reproduces itself. And what happens during those different phases of the cell cycle. Mitosis and meiosis are two processes of cell division that, once again, you should be able to compare and contrast with one another. They differ in terms of the number of chromosome numbers and genetic diversity of the daughter cells that they produce, and they have different functions. One of these processes is involved in what we call somatic growth. When you were conceived, when your father fertilized your mother's egg, you were a single cell, and you've grown into an individual with trillions of cells. And so mitosis is the process of all that cell reproduction that makes you into who you are. And then meiosis is how we make gametes for reproduction. So we'll talk about those differences. And then you need to know this information well enough because it serves as a bridge to understanding genetics. If we didn't need to understand genetics, then I wouldn't teach you anything about mitosis and meiosis possibly. But we need to understand genetics because, once again, Michael Pollan is going to talk repeatedly about genetics and genetic engineering over the course of the next couple of chapters. So that's kind of the goal for today, at least. So flowers. Flowers are difficult, I think, sometimes for animals to think about, and we're animals, and so we don't think about flowers as easily, because oftentimes flowers are what we refer to as complete. A complete flower is a flower that has both male parts and female parts. And so over here, this is a carpel on the right-hand side, and this is a stamen on the left-hand side. The stamen, or the staminate parts of the flower, are the male parts of the flower, and the carpel is the female part of the flower. And so male and female is a tricky concept in biology. And what's tricky about it is that it all has to do with the gametes that are produced. So these are the parts of the flower. They're right here at the middle of the flower. These are the parts of the flower that produce the gametes. The gametes are the sex cells of the individual. And in biology, the male and female is always determined by the size of the gametes. Females generally produce large, non-mobile gametes, and males produce small or even tiny mobile gametes. And so when we generally assign sex to an individual in any species, whether it's a plant or whether it's an animal, we refer to the male individual as the individual that produces tiny mobile gametes, and the female which produces larger immobile gametes. And so what happens in a flower, many flowers in the world produce flowers that have both male and female parts. And what happens is the anther, which is this large structure at the end of the stamen. So the stamen is made up of a filament and an anther. Inside the anther are pollen grains that are the results of meiosis. And these pollen grains are going to be released by the plant, and they will travel, they're the mobile gamete, they will travel to the stigma of the carpel. They will burrow down through the flesh of the stigma. They will then seek out the ovule, and they will fertilize the ovule. The ovule is just a fancy term for an egg. So if we were going to think about this in terms of animals, the anther produces the sperm, the carpel produces the egg, and the sperm, the pollen, fertilizes the egg. So in that way, it's analogous to animal systems, except that in this case, a single flower can produce both male gametes and female gametes. That's not always the case. So for example, in corn, male and female flowers occur on the same plant, but the female flowers occur low down on the plant, and the male flowers occur at the top of the plant. And you'll, if you've read Michael Pollan's chapter, he talks about the tassel at the top of the plant. The tassel are the staminate flowers, the male flowers, and the female flowers are the flowers that basically produce the corn cob, essentially. Those are the eggs that are going to be fertilized by the pollen. And so in some cases, the male and female parts of the flower are separated into different flowers on the same plant. In other cases, some plants are female and other plants are male. And so in the case of ginkgo trees, one tree will be female and produce flowers that only possess carpels. Another tree will produce flowers that only produce stamens. And so you actually have to have pollination occurring from one individual to another individual in order for reproduction to occur. One of the advantages of having male and female parts on the same plant is you can sometimes get self-pollination. So this plant could pollinate itself. In a lot of cases, though, selfing is precluded. And so in a great many flower species, the stamens of the same plant cannot pollinate the carpel of the plant that the stamen resides on. And so there are genetic mechanisms by which plants prevent selfing, because if you constantly self, you basically become an inbred population, and that's not good from a genetic standpoint. We'll talk about inbreeding when we get to genetics. But so any questions about basic flower structure? I don't really care about sepals and petals and things like this because those are less important than the stamen and the carpel. Okay, so when you look inside of a carpel, you see that there are a bunch of ovules inside of here. So this is a carpel with 3 divisions, and inside of each division there are ovules. Here's a carpel with only 1 division, and once again a bunch of ovules. And just depending upon the type of plant that you're looking at, this looks like it's a okra or something like that that has these internal divisions. Whenever you chop open a tomato, you can see the internal divisions that occur as a result of these internal divisions of the carpel. So a grass flower, it's incomplete because it doesn't have petals essentially, but oh, and I used the term complete and incomplete incorrectly earlier. It's perfect because it has both male and female parts. So once again, here are the anthers at the ends of the stamens, and then this is the carpel here. So it's a perfect plant because it has male and female parts, but it's incomplete because it's lacking some floral structure. So this would be both a complete plant because it has all the structures. It's got sepals, it's got petals, it's got the male and female parts, but it's also perfect because it has male and female parts. So when we think about the life cycle of a plant, I told you that when an organism has her eggs fertilized, it becomes a zygote, a single-celled organism. The single-celled organism will go through somatic growth. It will grow up and become an adult plant. Once it is an adult plant, it will go through the process of meiosis, And in meiosis, it will produce both males and female gametes. And these gametes will then be used to fertilize and repeat the cycle. And so in order to have gametes, you have to go through the process of meiosis. So meiosis is the process by which we form gametes, and mitosis is the process by which we go from being a zygote to being an adult plant. And so understanding those two things are, those two differences between mitosis and meiosis are important. So in order to understand all of this, we have to get into the weeds a little bit. So how many of you have heard of DNA in the past? Everybody? Heard of DNA? Why can I not see all of you? There we go. All right. So DNA is a double-stranded molecule, and it's made up of a phosphate molecule, a 5-carbon sugar, and these things called nitrogenous bases. So the red and the yellow and the green and the blue, these are all nitrogenous bases. And so it occurs in 2 strands. Each strand has a phosphate sugar backbone. So we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Then we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then we have these nitrogenous bases that are bound to the sugar. And then this strand on the left-hand side is bound to the sugar on the right-hand side through what we call hydrogen bonds. And I was going through this earlier, and I was like, why did I stick a picture of the, periodic table in here? And now I remember why I stuck a picture of the periodic table in here. And it's because it's useful at this point. So I'm going to go and move that back into place and go back to the slideshow. So these hydrogen bonds that are illustrated by little dotted lines are formed because oxygen loves to attract electrons to itself. So if you look at a periodic table of the elements, you see that there are these elements up here in the upper right-hand corner of the periodic table. Things like oxygen, nitrogen, sulfur, fluorine, chlorine. These are all atoms that are referred to as highly electronegative. And I don't know how many of you have taken chemistry in the past, but electronegativity is the tendency for an atom to want to pull electrons away from other atoms. And so you see that oxygen is up here right next to fluorine, and sulfur is up here right next to chlorine. Chlorine is so electronegative that it actually will pull electrons all the way away from sodium to give it a negative charge and give sodium a positive charge, and that's where we get table salt. So table salt is sodium chloride, and it's an ion. When chlorine and sodium interact with one another, chlorine pulls an electron away from sodium, which makes chlorine have a full negative charge, and then sodium has lost that electron, so it's got a full positive charge, and then those opposite charges cause them to bond together in what we call an ionic bond. Well, oxygen and sulfur have similar properties, but not quite as strong as fluorine and chlorine. And so when you have this oxygen, it tends to pull electrons away from this NH2 group, electrons away from the hydrogen actually. And it makes the oxygen partially positive and the hydrogen the oxygen partially negative and the hydrogen partially positive. And then that small difference in charge causes those two molecules to bond together through what we call hydrogen bonding. So similar thing happens in water. You guys might know that water is made up of an oxygen and 2 hydrogens. And one of the ways that you can think about electrons, so you guys remember your basic structure of an atom? What are the different parts of an atom? Three parts of an atom.
Protons, neutrons, and electrons.
Okay, so where do protons and neutrons live in an atom?
Nucleus.
What's that?
The nucleus.
In the nucleus, and then the electrons are found not in the nucleus. Well, if we look at this the way Niels Bohr drew these, he drew the atom as a nucleus, and then he drew electrons as occupying these shells outside the nucleus. And these electrons exist in pairs. And so the nucleus is here. There are 2 electrons in this first shell. There are 8 electrons in the second shell, et cetera, et cetera. Well, hydrogen just has 1 proton, 1 neutron, and 1 electron. Oxygen has 8 protons, 8 neutrons, and it has 8 electrons. So the first 2 electrons go into that first shell. Then the second shell has 1, 2, 3, 4, 5, 6 electrons in it. And so it has 2 electrons that are in unfilled orbitals. These things are called orbitals. And hydrogen has 1 unfilled orbital, and this allows hydrogen to bond with oxygen right here. And so you could also have another hydrogen bonding with oxygen right here. But when they bond together, oxygen tends to pull hydrogen's electron away from hydrogen. And so a more productive way of thinking about oxygen and hydrogen is to think about them as having a nucleus. And rather than putting these electrons into these static shells, think of electrons as little tiny particles. They weigh hardly anything at all. Little tiny particles that are just zipping around in space. So around this nucleus, electrons are just zooming around. And they form this cloud around the nucleus. The 1 electron of hydrogen is zooming around hydrogen, forming this electron cloud. Same thing is happening with this hydrogen over here, zooming around in space. Well, when hydrogen gets close to oxygen, oxygen tends to pull those electrons away from hydrogen such that this cloud, rather than hanging out all around hydrogen's nucleus, it gets pulled over. If this is the nucleus of hydrogen, rather than zooming around hydrogen, it tends to zoom around a little ways away from hydrogen's cloud, which means that oxygen becomes a little more negative and hydrogen becomes a little more positive. So when we talk about electronegativity, it's the tendency of an atom to pull this electron away from another atom and bring it closer to your own nucleus. Because that electron is negatively charged, it makes you a little more negative, and it makes the atom that you're pulling that electron away from a little more positive. Now, if we have another oxygen over here that is bonded to hydrogen, this partially negative oxygen can form a hydrogen bond with that partially positive hydrogen. And so that's what's happening in DNA. We have these weak intermolecular forces that are hydrogen bonds, but there are so many of them that it serves to basically connect the 2 strands of DNA with one another. And the whole reason that this happens is because oxygen tends to pull electrons away from hydrogen. And it's because it's in this group of highly electronegative atoms that are at the upper right-hand corner of the periodic table of the elements. So that's an innate characteristic of these types of atoms. As you get more to the middle, they tend to not do that. And they really don't do that over here. So, really complicated structure. Just think of it as two strands that are held together by hydrogen bonds in the middle. But the order of these nitrogenous bases becomes very important from the standpoint of genetics. And we'll get to that actually on Friday. So the DNA is actually found in these really long strands, and the DNA ends up getting wrapped around proteins called histone proteins, and then these histone proteins get all bundled together in long bundles and ultimately become chromosomes. And so in eukaryotic organisms, these chromosomes are linear, and they're basically built out of DNA wrapped around protein molecules, all bundled together in these big, thick ropes. You can use heat to basically break the bond of the DNA with the histones, and you can ultimately use heat to split the two parts of the DNA molecule apart. We use heat to do that typically, but then the cell itself has enzymes that go about doing this. So remember, the enzymes lower the amount of energy that you need in order to get a chemical reaction to go to completion. And so while when we manipulate DNA, we'll oftentimes use heat, the cells themselves do this manipulation with DNA, with using enzymes of various types. So DNA wrapped around histones, histones bundled together into chromosomes. And when we think about mitosis and meiosis, we're going to be thinking about, well, what happens to these chromosomes during mitosis and meiosis? So when we think about chromosomes, we have to think about all of the chromosomes in the genome of an organism. So in humans, we have 46 chromosomes. So this is Paul, right here. I come from two parents. My father is named Philip, and my mother is named Julia. My mom had 46 chromosomes, and my dad had 46 chromosomes, and I have 46 chromosomes. So my 46 chromosomes, half of them came from my mom, and half of them came from my dad. So I got 23 chromosomes from my dad, and I got 23 chromosomes from my mom. And at fertilization, her 23 chromosomes came together with my dad's 23 chromosomes and gave me 46 chromosomes. So in this karyotype, what we have is 23 different kinds of chromosomes. Chromosome 1, chromosome 2, chromosome 3, chromosome 4, all the way down to chromosome X here. So 23 different types, 22 what we call autosomal chromosomes and 1 sex chromosome. One of these chromosomes came from your male parent, the other chromosome came from your female parent. One of these came from the female parent, the other one came from the male parent. Male, female, female, male. I can't tell from looking at these which one came from which parent because they basically look exactly the same because they're the same type of chromosome. See how chromosome 1 is the same length, both from the male and the female. Chromosome 3 is a different length. Chromosome 4 is similar in length to chromosome 3, but this little dark band in the middle here, which is called a centromere, is in the middle here, whereas it's close to one end in this chromosome. So chromosomes differ structurally in terms of their length and where this thing called the centromere is located. So if you've been paying attention, you might have noticed that this chromosome has 2 arms, whereas these chromosomes each only have a single arm. Well, this chromosome has 2 arms because it has duplicated itself. This chromosome has a single arm each on each of these chromosomes because it hasn't gone through this process of duplication yet. So if we look at the human karyotype, you can stain chromosomes with a particular stain that allows you to see banding patterns in the chromosomes. And you'll see that each chromosome has its own unique kind of banding pattern. And you can see right here, there's a section where the chromosome looks constricted in this drawing. And that place right there is the centromere. And you see that in some chromosomes, the centromere is located kind of close to the middle of the chromosome. But then in other chromosomes, the centromere, like on chromosome 7 here, the centromere is placed nearer one end than the other end. And in some cases, the chromosome is right out at the very tip of the chromosome. Centromeres are places where the chromosome is really densely packed. And as a result, it tends to not take up stain very well, which is why oftentimes in these kinds of images, you see the centromere is basically just not visualized because the chromatin, the mixture of the histones with the DNA, which is what is called chromatin, that is so densely packed at the centromere that the dye that leads to these chromosomes glowing doesn't penetrate, and it can't bind to anything in there. And so you can see where the centromere is because it doesn't appear. It just simply doesn't show up when you've stained chromosomes this way. So a karyotype is just a visualization of the chromosomes in an organism. And so this is what it would look like under a particular kind of staining. And then we can basically separate the chromosomes by their lengths and also by their common banding pattern. And so there is a DNA molecule that is snaking around the entire length of chromosome 1, but it has a beginning and it has an end, and it's all one long linear molecule down the entire length of that chromosome. So the staining depends on how densely packed. And I guess here the staining is dark is densely packed chromosome and light is less densely packed chromosome. I had it opposite in my head. So if we take these chromosomes and we look at their banding pattern and get them grouped together, we see that there are 2 examples of chromosome 1, there are 2 examples of chromosome 2, 2 examples of chromosome 3, chromosome 4, and these dotted lines represent where the centromeres are. This is before the chromosome has duplicated itself. This is after the chromosome has duplicated itself. So now chromosome 1 has 2 members. One came from your mom, one came from your dad, but the one from your mom has duplicated itself, so now that there are 2 copies of it. The one from your dad has duplicated itself so that there's now 2 copies of it. And so both this single unit here is referred to a chromosome, but also this unit here that has 2 arms is also a chromosome. And this is one of the places where both CTI 259 students and my genetic students and my Bio 133 students get confused. So I want to clarify the terminology here. So if this is chromosome 1, this is a chromosome that has not undergone duplication yet, and we would refer to this as a chromosome. As the chromosome prepares for cell division, it's going to duplicate itself. So it's going to create a new copy of chromosome 1 that is attached to the old copy of chromosome 1 here at the centromere. And at this point, we would call this a single chromosome with 2 sister chromatids. And one of these came from your mom and the other came from your dad. And so if we stained these with, I think that's Giemsa staining, we would see that the same banding pattern occurs on all of these different copies of the chromosomes. But this is the original copy of your mom's chromosome on the far left-hand side. This is the new copy of your mom's chromosome on the right-hand side of this first chromosome. This is your dad's chromosome originally. This is the new copy of your dad's chromosome, but you see that they're exactly the same in terms of their banding pattern. Not only that, all of the individual variations in the genes from your mom that are on this original chromosome are also found on this duplicated chromosome, this duplicated chromatid. So we have one chromatid, two chromatids from your mom, one chromatid, two chromatids from your dad. So this is a chromosome. This whole thing is also a chromosome. This dark purple is one chromatid. The light purple is another chromatid. Chromosome from mom, chromosome from dad. Mom has two chromatids, dad has two chromatids, because it's duplicated the chromosomes as chromatids getting ready for cell division. Now, who is totally lost at the end of that explanation? Raise your hand if you are lost. So everybody's relatively clear about that? You guys should all become biology majors. You guys are so quiet. Okay. Then if we're clear on that, let's go to this. So how do we get from this single chromosome to a chromosome with two chromatids? Well, one of the things that we have to do is we have to make copies of the DNA. And so this is called DNA replication. This is another way of thinking about the DNA, where this ribbon is the sugar phosphate backbone of the DNA molecule. And then these little ribbons in the middle are the nitrogenous bases. Remember that these nitrogenous bases are held together by a hydrogen bond. And so it's really pretty easy to split this DNA molecule into the two halves. Once you've split it into the two halves, you can basically take new DNA nucleotides. So I didn't give you this term, and I probably should have back then. A nucleotide is made up of a phosphate molecule bonded to this 5-carbon sugar that is bonded to a nitrogenous base. These are the repeating units of DNA. And then to get this molecule of DNA longer, you take another phosphate that is bonded to a sugar that is bonded to a nitrogenous base, and you bond that phosphate to that sugar, and that begins to make this long chain. So what happens in DNA replication is you basically take this double-stranded molecule, you split it into two halves, and then on the nitrogenous bases, you add new nucleotides to the open nucleotides that are on there. And you do this through what we call complementary base pairing. And complementary base pairing is simply the process by which there are 4 different nitrogenous bases. Adenine always bonds to thymine, and cytosine always bonds to guanine. And so if you unzip this DNA molecule and you expose an adenine on one side of the molecule, then the cell knows that a thymine needs to go onto the adenine. If you expose a cytosine, then a guanine will attach to that. So you see that everywhere you see a G in this model, there's a C attached to it. Everywhere you see a T, there's an A attached to it. So AGTC, those things refer to adenine, thymine, guanine, and cytosine. You guys ever seen the movie Gattaca? No, nobody's watched Gattaca. It's a movie that starred Ethan Hawke and Uma Thurman and Jude Law, and it's about this dystopian future where everybody gets their genes sequenced when you're a baby, and if you don't like your baby's genetic code, you can just go in using genetic engineering technology and change what your baby is going to be like, and so they can genetically engineer babies. And what role you play in society is ultimately determined by your genetic predetermination. So it's a very dystopian sort of place, and it's called Gattaca. Because it's essentially a name of a movie that is used from, that is derived from guanine, adenine, thymine, adenine, cytosine, adenine. That's where the name comes from. So, so this complementary base pairing is how the cell keeps straight which nucleotides get bonded to which other nucleotides as it forms a new molecule of DNA. And so the blue ribbon strand here is the original DNA. The pink ribbon strand here is the new DNA that is added to the old DNA. So every new strand of DNA has some of the old DNA in place with some new DNA added to it. And this is why it's called semi-conservative replication. It always uses old DNA as a template for creating new DNA sequences. Okay, does that make sense? If you're in a biology class, we would go through all the enzymes and everything that are involved in all of this, but we're not going to do that because this is not a biology class. You just need to know that DNA can be replicated from old DNA, and you have to do this before you get ready to go through cell division. So the cell cycle is the way in which we envision the life of a cell. Cells come about through the process of mitosis. After mitosis forms new cells, the cells go through a growth phase, which is called G1. Then they go through a synthesis phase, which is where they synthesize new DNA. So you see, we went from a chromosome with only one chromatid to a chromosome with two chromatids. It's because S refers to synthesis. This is the synthesis phase. And the main thing you guys need to worry about is that during that synthesis phase, DNA is replicated. And so we go from chromosomes that look like this to chromosomes that look like this, because now there's two copies of the DNA. And then the G2 phase is just a further growth phase where the cell is preparing to divide. And then when they divide, that phase is called mitosis, which produces two new daughter cells, which then start the cycle all over again. Whether you're going to divide through mitosis or through meiosis, you have to go through this S phase in order for the cell to have enough copies of DNA to go through the process of cell division. Okay? So the synthesis phase is really crucially important here. Okay, mitosis. Don't be frightened by all of the busyness on the slide, because what you need to do is you need to go through it step by step. So let me just orient you here at the beginning. Here is a cell, and this is an animal cell in this case, and I realize we need to talk about plant cells, which we will here in a little while. But the cell has a nucleus, and inside the nucleus is where the chromosomes are. And so at the very beginning of, well, in the S phase, we go from chromosomes that are single copies to chromosomes that are two copies of one another, a chromosome with two sister chromatids. So that happens during the S phase of the cell cycle. But once that happens, the beginning of mitosis involves a couple of different things happening. In this phase called prophase, so you will need to know these different phases and some of the major things that happen during them. So in prophase, there is normally a membrane that surrounds the nucleus and keeps the genetic material, the chromosome, separate from the cytoplasm, the other parts of the cell. So the nucleus is a membrane-bound organelle that possesses the genetic code of the cell. During prophase, that membrane starts to break down so that there's no longer any separation between the cytoplasm of the cell and the chromosomes. And the other thing that happens during prophase is that these chromosomes that were in really long, thin, strung out chromosomes have contracted on themselves and become thicker and shorter. They've become more dense as a result, which is why you can begin to see them in these two-armed versions of themselves. So in prophase, the nuclear membrane breaks down. And the chromosomes themselves do what we call condense. Condense is like condensing milk to make concentrated milk. You basically boil it down until it's a much stronger, much more concentrated version of itself. So these chromosomes that were in these really long strings during the S phase and the G phases of the cell cycle, they become shorter, and as a result of becoming shorter, they become thicker. And so this condensation occurs. And then the other thing is that there are organelles that begin to migrate to opposite sides of the cell. These things are called centrioles. You don't really need to know them. But there's this migration of parts within the cell. Now, the next phase, which is metaphase, is best characterized by this thing called the metaphase plate. As these chromosomes are thickening during prophase, there are also other things happening in that there are microtubules called spindles, and these spindle fibers form and attach to the centromere of each chromosome. And so remember, the centromere is this dense chromatin in the middle, and there is a spindle fiber that makes a physical connection to the chromosome. And as a result of that, because the spindle fibers come from each side of the chromosome, it positions all of the chromosomes along the middle of the cell. And if you turn that middle of the cell on its side, it would look like a plate of chromosomes. And that's actually how we get karyotypes like this, is we catch cells during metaphase where we can see all of the chromosomes in their thickened form, and then that's how we get the karyotype. And so in metaphase, these duplicated chromosomes all lie in a common plane across the cell. They're called the metaphase plate. Then in anaphase, which is down here in the lower left-hand corner, what happens is the two chromatids become separated, and they become separated at the centromere. So going back to this situation here, the two chromatids are held together at the centromere, and in anaphase, this chromatid is going to become separated from this chromatid, and they will start to be drugged to the new parts of the new cells. So in anaphase, what you begin to see is the two chromatids separating and pulling apart from one another. So the key thing to remember for anaphase is that these chromatids separate. Now, in telophase, these chromosomes that used to be chromatids, these chromosomes are now found in newly developing nuclei. And in a thing called cytokinesis, the cell begins to separate its cytoplasm until at the end of mitosis, you now have two new cells. Each new cell has its own chromosomes and is totally separated from the other chromosomes. So telophase is characterized by the formation of new nuclei and the onset of cytoplasmic division. And the fancy pants term for cytoplasmic division is cytokinesis. But look what's happened here. How many chromosomes do we have at the beginning of mitosis? Blair?
4.
We have 4. We have 2 copies of the red and 2 copies of the blue. One of the red one came from the mom of this the organism that possesses the cell. One came from the dad. One of the blue ones came from the mom. One of the blue ones came from the dad. So we have 4 chromosomes here. How many chromatids do we have here? Here at prophase. 4 chromosomes. Each chromosome has how many chromatids?
2, so it's 8 total.
So we have 8 chromatids. Who was that? Mackenzie?
Yes.
So if we have 8 chromatids, how many copies of DNA do we have?
Would it just double it again?
Nope, because each chromatid has a copy of DNA along its length. So if we go back to this, there's 1 copy of DNA snaking along this chromosome here. When that chromosome duplicates, now we have 1 chromosome with 2 chromatids. Each chromatid has a copy of DNA on it. So we have 1 chromosome, 2 chromatids, 2 copies of DNA. So at this point, we have 4 chromosomes, 8 chromatids, 8 copies of DNA. At this point, those 2 copies of DNA get separated. So on this chromosome that's moving to this side of the cell, we have 1 copy of DNA here, another copy of DNA here. 1 copy of DNA here, another copy of DNA here. 1 DNA here and its copy here. So that when we get to telophase, how many chromosomes are in this new cell being formed?
4.
4 chromosomes. How many chromatids are supposed to be in that cell?
It's supposed to be 8, right?
No, there's only supposed to be 1, 2, 3, 4. This is a figure from a textbook. Guess what? Somebody messed up because these are not supposed to be duplicated chromosomes. These are supposed to be single chromatid chromosomes. This is a mistake from a textbook figure. These should only have 1 branch for each of these. There should be 4 chromosomes and only 4 copies of DNA in those. Because at the end of telophase, when you have 2 new cells, there's only 1 chromatid on each of these things. So this is a mistake. These things right here should look like these things right here, and these things are wrong. The textbook authors made a mistake here. So correct that in your notes. These should only have 1 copy of each chromosome here, not 2 copies like this. So mitosis takes cells with 4 chromosomes and ends up with cells with 4 chromosomes. And these chromosomes are genetically identical to the chromosomes in the original cells. And so if we're going to think about comparing mitosis and meiosis, we can think about it from a variety of different perspectives. one way we can think of it is in terms of the number of chromosomes that we start with. And we start with 2n, or what we call diploid number of chromosomes. Diploid basically goes back to thinking about this karyotype. We have 23 different kinds of chromosomes, but we have 2 copies of each. One copy came from your mom, one copy came from your dad. And so we have 2 copies of 23 chromosomes. 23 different kinds of chromosomes, that's what the N is, and we have 2 copies of each. This is diploid. So in this example here, we actually have a diploid number of 4 chromosomes because we have 2 copies of 2 different kinds of chromosomes, the red chromosomes and the blue chromosomes. So in this case, the diploid number is 4, but it varies from species to species. We only go through one phase of cell division. This is one phase of cell division where we go from 1 cell to 2 cells in the end. The number of daughter cells that we have at the end of this is 2 daughter cells, and the number of chromosomes that we have in those daughter cells is diploid, just like it was at the beginning, and these chromosomes are all genetically identical. So one of the ways that you can think about mitosis and meiosis is to put it in a table like this to get across what the similarities and differences are between them. And so then we have to ask ourselves, well, how does meiosis differ from mitosis in terms of what's the ploidy? This deals with ploidy, and you can be once again, you can be either diploid, which is 2n, or you can be haploid, which is 1n. One copy of the however many different kinds of chromosomes you have. So I think we're just about out of time. So what we'll do is we will look at meiosis tomorrow. That's the next thing in the slides. Go and read about meiosis in your textbook if you haven't already, and we will go through the phases of meiosis in some detail, and we'll then continue filling out this chart that compares and contrasts these 2 forms of cell division. Questions? I could try and cram it in the next 5 minutes, but I would just go through it too fast and confuse you more than anything else, probably. Questions about mitosis so far? All right. Well, the next slide here. So this is it in animal cells, but you can actually see this with a simple microscope. If we had a lab with this class, I would take you up to lab, and I would have you look at this in a microscope. This is the onion root tip, and at the root tip, remember that's where cells are undergoing cell division because that's where the root is growing. And what you can see in this is an actual photomicrograph of an actual root. And there are places where you see that the chromosomes are lined up along a line across the middle of the cell. Here, 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 here. These are places where you're seeing metaphase because those chromosomes are at the metaphase plate. You can see interphase, which is just the cell cycle. This is where the cells are just kind of normally resting and growing and synthesizing new DNA. They just look like normal cells. You can see situations where the chromosomes are being pulled apart, being pulled to opposite sides of the cells. And then you can see places where those chromosomes have started to form new nuclei and you can see a new cell membrane and cell wall forming to separate this new cell from that new cell. That's the parts of telophase. So in all of these cases, you can see chromosomes condensing, which would be prophase. You can see metaphase, where they're lined up along the metaphase plate. You can see anaphase, where the chromosomes are being pulled apart into new cells. And then you can see the formation of new cells in telophase. And this is exactly what it would look like if we were looking at it under a microscope in lab. And that concludes mitosis. Like I said, we'll cover meiosis on Friday. All right, look over this stuff between now and Friday and come to me with questions at the beginning of class on Friday about anything we've covered so far. All right, I'll see you on Friday.